In previous chapters, we covered the structure of DNA in terms of the actual nucleotides required to build the molecule, as well as the related but different nucleotides needed to build RNA. In Chapter 11, we're going to look at how do cells use their DNA and RNA in their genetics. And in particular, we're going to look at how does DNA make a copy of itself, how does the cell use the DNA to get RNA, and then how does the cell use that RNA as the instruction manual for building proteins. This image shows us E. coli and elephants, two beings that begin with the letter E. E. coli are, of course, bacteria and are prokaryotic. They do not have a nucleus. And elephants are animals, so they are eukaryotic. And of course, their cells do have a nucleus. Despite their dramatic differences in cell structure and appearances, the very basics of the DNA, the chemicals that compose that DNA, are pretty much identical. DNA is always made of the deoxyribose connected to a phosphate, and on the other side at the first carbon connected to some nitrogenous base, the adenine, cytosine, thymine, or guanine. No matter what the organism is, or the virus, the DNA is structured exactly the same, the DNA molecule. The chromosomes of a prokaryotic cell, each one having a single circular chromosome, are put together differently than eukaryotic chromosomes that are linear, and we have multiple chromosomes. But the chemical DNA that makes up either structure of a chromosome is chemically indistinguishable. The flow of information in cells, and I did underline in cells because the flow of information in viruses doesn't necessarily follow this process. We're going to stick to cells in this chapter. The flow of information begins with DNA in the nucleus of a eukaryotic cell or the nucleoid of a prokaryotic cell. We can make a copy of DNA through replication, but if we're going to get out of that DNA information into the instructions provided to the cell, we have to undergo the process called transcription, where an enzyme will read the DNA and create RNA. Both of those are nucleic acids, but this is deoxyribonucleic acid, and RNA is ribonucleic acid. The DNA does not get changed by this process permanently. It's temporarily opened up and the enzyme reads the strands, but it is returned to its completely functional state. It does not get changed into RNA. The DNA remains. So after transcription, we will still have the DNA, but we will also have RNA. And transcription is used to produce the three different types of RNA: messenger, transfer, and ribosomal. Which type of RNA gets made depends on what is the DNA sequence that the enzyme reads. And then we go from RNA, we need the process of translation to have our messenger RNA read by the ribosomes, providing the instructions for linking amino acids together through dehydration synthesis to build a protein. And I've labeled above here nucleic acid, and then new category. Because one way to remember transcription versus translation, since they both have trans in their prefix, meaning across from, transcription is taking the language of the nucleic acid DNA and transcribing it into the slightly different but related nucleic acid of RNA. So this would be, say, written English and I read it to you the exact same words, but the format is different. One format is written, one format is oral, but the words are exactly the same. Translation takes the language of nucleic acid and creates a whole new language, protein, a different category of molecule. So English to Arabic, Hebrew to German, Italian to Russian. Same information, but a whole new format, completely different category of it, translation. In cells, this is always what we need. If we're going to get a protein, we have to originally have the genetic information in the form of DNA. We can't go directly from DNA to protein. The cell has to have enzymes that will read that DNA information and create an RNA-related copy. And then the ribosomes will translate genetic information of RNA into proteins that will do work in the cell. This is showing that even though we could have the exact same sequence of nucleotides, the genotype, what genes are present in our genome, one environmental condition will produce a particular phenotype or the physical characteristics. That same organism with the same genotype under different environmental conditions might have a different phenotype. This is particularly true of our prokaryotic cells. They have only one chromosome, and if they find themselves in different environmental conditions, they need to be able to adapt and can express different characteristics based on the environment. This slide shows what were originally three concepts of how DNA gets copied in cells. There is the conservative, meaning that the original sequence gets conserved during the process of replication, where we have two original strands, and after they're copied, the original strands come back together and two entirely new strands are made, so that if the organism that had this DNA replicated again, these two new strands would show up, and we'd make these two old strands would be copied and they'd stay together, and we get two new strands, and then the reds, we would get only red strands out of that. Semi-conservative means each new strand conserves one of the original strands, but gets a new strand. So that if this molecule gets replicated, this strand is that one, and it gets a new strand, and this strand is here, and it gets a new strand. Dispersive says that when this molecule replicates, each subsequent new molecule is a mixture of original and new DNA. The semi-conservative model is the one that in fact happens inside cells. And there were many experiments to be able to demonstrate that, and we will look at one here. This used an isotope of nitrogen. It says heavy 15N. Non-radioactive nitrogen has a mass of 14, and so nitrogen 15 has one additional neutron. Having an additional neutron is what changes that mass. So Messelson and Stahl used E. coli because it's a simple microbe, it's very easy to cultivate in labs, and they let it grow in heavy nitrogen, meaning when its proteins, but in particular its DNA, was being replicated, the heavy nitrogen is the only nitrogen available to build new nucleotides, and so the DNA would be nice and heavy. As long as you allow it to replicate only in the presence of heavy nitrogen with no other nitrogen sources present, you end up with double-stranded DNA where all of the nitrogens have the heavy form. These tubes represent if the cells grown in the presence of this heavy nitrogen were put in a centrifuge, then we could see where the radioactivity is, and that will tell us the cells have either all heavy nitrogen or some sort of mix in the next steps. This row indicates that they allow the cells to undergo one round of replication. The scientists could determine how long does it take for E. coli to divide, allow that much time to pass when they take these cells and put them into a beaker that has only nitrogen-14, the other isotope, the lighter weight isotope, and allow that to spin, and what you see is each of the new strands has one blue original heavy strand and one red new light strand. If the replication was conservative, you would have half the cells keeping their original two blue strands and half the cells having two new lightweight strands. If it's semi-conservative, then after one replication, all of the cells would have this mixture of one heavy and one light strand. And if it's dispersive, well, everything gets mixed up. And so you wouldn't be able to tell the difference between these two forms of replication after only one round. So they allowed a second round of replication, still in the lightweight nitrogen, did a spin, and what they found was you end up with half of the cells have the mixture of heavy and light, and half have only the lightweight DNA. So we did not see that with the could not be conservative. If it were dispersive, we would still see all of the strands having a mixture of old and new, or heavy and light. What we did see is now we've got half of the cells containing the mixed, one heavy, one light, and half of them having only the new two rounds in lightweight nitrogen. And that was the evidence supporting that DNA replication is semi-conservative. Each round of replication means the strands separate, and each of those original strands gets one new strand along with it. This is a deoxyribonucleotide. It was missing an H, so I drew one in. That is the deoxyribose. There are the three phosphates, and this happens to be a guanosine because this is guanine. These static pictures of DNA replication aren't particularly useful. There's a lot of information. It tells you the names of the important structures and enzymes, but it doesn't really tell you, gracious, I need more coffee, how the replication is actually happening. We do have the two strands of DNA. They are not just completely unzipped and then replicated. They are opened up a little bit at a time, and that opening is carried out by this enzyme called helicase, because it is an enzyme. It acts on the helix to open it a little at a time, and that causes this what's called the replication fork, because the helicase splits open the fork. We will need DNA polymerase, an enzyme building a polymer, and the polymer is DNA. We are also going to need something called RNA primase. We have something called the lagging strand and a leading strand. The leading strand says it is continuous synthesis. Once the synthesis begins, it continues uninterrupted, being read by this DNA polymerase until we reach the end of the molecule. The lagging strand is called that because it cannot be replicated in a continuous motion. The reason for that is that this DNA polymerase can only add or build a new molecule from the 5 prime, the end with the phosphate, toward the 3 prime end of a molecule, the end with just the OH. And that means the template, the strand of DNA that is the instruction manual for DNA polymerase 3, has to have a 3 prime OH. 1, 2, 3. Third carbon, it has to have this. That is the shape that DNA polymerase 3 recognizes. If we don't have this on the end, DNA polymerase 3 can't recognize it. We have that on the leading strand, so the DNA polymerase 3 can bind and proceed. The lagging strand, this end is a 5 prime that has the phosphate on it. Wrong shape. DNA polymerase 3 can't bind to that. Instead, we have an enzyme called RNA primase. It builds a primer made of RNA. A primer is something that helps to get a reaction started. In this case, it is made of RNA. And this RNA primase synthesizes this little piece of RNA that is from the 5 prime to the 3 prime. That means there would be a 3 prime OH here that the DNA polymerase can bind to and build this little red piece of DNA that will be synthesizing in the 5 prime to 3 prime direction. And so each of these red fragments means RNA primer was put here, DNA polymerase 3 bound and made a piece of DNA. And then RNA primer was placed here, DNA polymerase 3 bound to the 3 prime end of the RNA and built this stretch. And we keep going as the fork opens further and further back until we complete it. So that's why it's called the lagging strand. We have to wait for the DNA to open a little bit, the primer to be put on, DNA polymerase 3 to bind, and then make a little piece. Then we have to jump back and make a primer and then bind and continue. Because we will have little pieces of RNA left, we then have DNA polymerase 1 will remove the RNA and replace it with DNA. Each of those little fragments was called an Okazaki fragment. There will be bits of RNA between the Okazaki fragments. DNA polymerase 1 removes the RNA, literally cuts it out, and then replaces it with DNA. And then any gaps that are left, that there's not a covalent bond in that sugar phosphate backbone, is then sealed with DNA ligase. To ligate means to seal up. So DNA ligase seals DNA and makes sure we have a continuous strand. I embedded a DNA replication video that I won't play all of it here, but these slides will also be saved in the module so that you can go to the slides without me talking over them and be able to play this video. It is captioned, is accurate, and goes through the leading and lagging strands and how the enzymes take care of that. Oop, did not mean to play it. Eukaryotic chromosomes are linear, and so we don't just start at one end and zip all the way to the other. The DNA helicase opens up several bubbles and then replication continues in both directions. Otherwise, it would take entirely too long for the DNA to copy. But we still end up with 2 strands, each of which has one original strand and one newly synthesized strand. Because of the linear chromosomes, eukaryotes have a problem where the ends of them might tend to get shorter. So we have telomeres at the ends of our chromosomes that are a repeat sequence, and it's called non-coding DNA, meaning it doesn't encode any sort of product. It's not junk DNA because we do need it. What it does is have a repeat sequence of nucleotides, and the telomerase or the telomerase, depending on who you ask, has a stretch of RNA in it that is complementary, can base pair to that repeat telomere sequence, and it extends the end of the chromosome, making sure that the part of the chromosome that is not the telomere does get completely replicated. There is a hypothesis called the telomere clock, where our telomerase functions less and less as we age, and that can result in the chromosomes getting shorter. Telomeres aren't an issue in prokaryotes because their DNA is a circle. There's not an end. Once the whole circle has been replicated, there's not any overhang left. This is a prokaryotic circular chromosome. We have some sort of little nick by the DNA polymerase III that will then cause the chromosome to open up and helicase can bind, and we get the replication of the DNA molecules so that we would still end up with one original and one new strand. And here, one original is on the outside and the new strand is on the inside. We still would have two genetically identical DNA strands, each of them having an original and a new strand. Transcription is taking the genetic information that is held within the DNA and encoding it and reading it to make an RNA copy. The DNA, again, is not changed permanently by this. It will seal up and be its whole original self, but we will have an RNA molecule carrying a single-strand copy of the information that was in the DNA. So the blue is representing DNA. We have the template strand, meaning that is the strand the polymerase, the RNA polymerase, is going to read, and the non-template strand, meaning this strand is not going to result in RNA. The RNA polymerase binds to the DNA, and it starts its binding at a sequence called the promoter. And that sequence is TATAAT. It is somehow pronounced the Tata box, but it is TATAAT. That is called a conserved sequence. Whether it's any gene in us, or our dogs, or E. coli, or an elephant, that particular arrangement of nucleotides is the signal for RNA polymerase to bind. We only need the RNA polymerase. It carries the information necessary to break the hydrogen bonds of the DNA in a localized area and to catalyze the synthesis of the RNA. We use the DNA sequence, and the RNA polymerase will add the RNA nucleotide that is complementary to the DNA. So if the DNA has an A, the RNA will have a U, uracil, because RNA does not have thymine. A's in DNA direct a U in the RNA, G's direct C's, T's direct A's, because T's pair with adenine, and RNA does have adenine, C's and G's. So CG pairs don't change, T in the DNA does result in an A in the RNA, but an A in the DNA results in a U in the RNA. And as the RNA polymerase moves along, there will eventually be a signal that indicates we've reached the end of where that RNA polymerase needs to read, and the enzyme will fall off, and the DNA seals back up behind it. Once we have our RNAs, then the ribosome has to be able to read that information and decode it to make the proteins. This chart is called a codon chart, and a codon is a triplet of nucleotides in the messenger RNA, and that will tell the ribosome and the tRNA which amino acid needs to be delivered. And we have 4 first letters, 4 second letters, 4 third letters. So there are 4 nucleotides. There are first, second, and third positions. So 4 to the third power is 4 times 4, which is 16. And 16 times 4 is 64. I did the math before I started talking, so it's right. There are 64 codons in here, 64 unique 3-letter combinations based on the 4 possible nucleotides. We do have 1 that's in green and 3 that are in red. The green one, AUG, will direct the cell to deliver the amino acid methionine, abbreviated here as MET. But UAA, UAG, and UGA do not have an amino acid. It just says stop. If the messenger RNA sequence is one of those, that is the signal to the ribosome and the tRNA that we have reached the end of the sequence that dictates the production of a protein. When we look at the possible combinations, there's only one combination of letters of nucleotides that results in a methionine. That is the start signal. But there are 6 combinations that give us this amino acid, serine. There are 6 combinations that give us leucine. There are 6 combinations that give us arginine. Each 3-letter combination can only result in a single amino acid. It must be the specific signal, the specific message for only that amino acid. Otherwise, it would be random which amino acid gets added. However, a given amino acid might have more than one genetic code to get there. Methionine only has 1 because it is the start signal. How does the ribosome know we start here? We want one way to do that. We don't want it to have many possible options because we want to be precise in building our protein. The amino acids that have the most codons are usually found in the active sites of enzymes, meaning it's really important that we get the right amino acid in that position. You see here, if it's CU, we could have a U, a C, an A, or a G. As long as the first 2 letters are correct, you're still going to get the right amino acid in that position. And then we also have a couple of other options for that leucine. That ability to have more than one codon still get you the right amino acid is called wobble. The code wobbles. And that helps with preserving function, because the proteins are carrying out the functions in our cells, even if there is a mutation. If there is some change in the DNA that results in a change in the RNA, but we end up with the same amino acid, then our genetics has been a little wobbly. So we have to have this one codon cannot specify anything other than this amino acid, but this amino acid might have more than one way to arrive there. So it is beneficial to have more than one way to get the correct amino acid, because the amino acid sequence is the primary structure, but that dictates the secondary and tertiary structures, and that tertiary and possibly quaternary structure dictates function. If we mess up the primary sequence, we might mess up the function. So we want to make sure we get that primary sequence right, and that happens even in the face of mutation. So in prokaryotes, we can have a lot of RNA polymerases going on a single gene at the same time. So this RNA polymerase 1, 2, 3, and 4, and they are making the messenger RNA, and as soon as the messenger RNA is made, the ribosomes can attach and start making protein. Because all of this is happening in the cytoplasm. There's no other place for it to happen. In eukaryotes, the RNA gets made in the nucleus, but it has to exit the nucleus before it can be translated. So one RNA polymerase reads the DNA and makes one RNA that exits the nucleus and then gets translated. Prokaryotes don't have to wait. Their whole DNA to RNA to protein is a lot more efficient. This is showing our messenger RNA as linear from 5' to 3'. This is our tRNA. It is a single-stranded molecule, but it does fold on itself with 4 areas where there can be hydrogen bonding. And the 3' end is where the amino acids will attach to get delivered to translation. So 3 different ways to show this, very cartoony way. This is a molecular model, and then here we just show a super stripped down cartoon of it. We're going to look at translation in bacteria because there are not any complicating factors. I know that sounds like a ridiculous thing to say when there's so much on here, but we really just have the messenger RNA, the transfer RNA, and the ribosome interacting with amino acids. Here is our messenger RNA. The codons are on the messenger RNA. The first thing that has to happen is the small ribosomal subunit recognizes that there is a start codon. The tRNA with the first amino acid, which is going to be methionine, because if there's AUG, that is the codon for methionine. That means the tRNA that has the complementary sequence called the anticodon. mRNA has the codon, tRNA has the anticodon. Because of the anticodon, that dictates which amino acid will be attached at its 3' end. The tRNA binds to the messenger RNA, so there is hydrogen bonds between the codon and the anticodon, and then the large ribosome makes the big sandwich over the top of this. We see these 3 sort of pale spaces. Those are actually little compartments within the ribosome, and they are called the E site, which is empty. It's also called the exit site. There is the P site, which is the peptide site. As this process continues, it will be holding the tRNA with the peptide connected amino acids connected by peptide bonds. And this site to the far right is called the A site, or the acceptor site. It accepts the tRNA with a single amino acid, waiting for the peptide bond to form. So once we initiate that first tRNA, and it has to be in the P site, even though there's not a peptide attached yet, because we need the acceptor site to be available. The next codon would tell us which tRNA with its amino acid would sit in the acceptor site. This arrow indicates that there would be a peptide bond formed between this amino acid and under the chain. The ribosome literally shifts one space, one codon over, and that means we would get, once this binds to there, we shift the peptide containing tRNA to this spot, and the empty tRNA would exit. This continues until the ribosome reaches a stop codon. There is no tRNA for a stop codon. Instead, a release factor binds, and that triggers the ribosome to come apart, the polypeptide to be released, so that would be the primary sequence, and then the protein could finish undergoing its secondary, tertiary, and possibly quaternary structures. So in prokaryotes, there is our single chromosome in the cytoplasm. We have transcription producing this pink messenger RNA, and multiple ribosomes can bind to it and do translation and get multiple copies of protein. In eukaryotes, we have the nucleus. Transcription happens in the nucleus. We have here labeled a pre-messenger RNA, and then we get a mature mRNA, and that mature mRNA has to leave the nucleus into the cytoplasm, where either free ribosomes in the cytoplasm can translate it, or it gets read by the ribosomes that are stuck to the surface of the endoplasmic reticulum. This mature RNA happens because in the DNA of eukaryotes, we have both coding, meaning it is a sequence of DNA that will be transcribed to RNA and translated to protein, and we have non-coding DNA. We saw the telomere as being an example of that, but we have non-coding DNA throughout the genome. Non-coding DNA gets transcribed all along with the coding RNA, but those areas that do not code for a protein are enzymatically cut out so that only the coding pieces of the RNA end up in the final messenger RNA, and we also give it a little cap called the 5' cap that chemically protects that end from being degraded, and we get a little tail on the 3' end to protect it from being degraded. Looking at the difference and similarities of translation, the ribosomes are different. You don't have to memorize how many RNAs, but the bacterial RNAs are smaller than the eukaryotic RNAs. The initial tRNA is methionine in eukaryotes. This F-met is formal, F-O-R-M-Y-L, formal methionine, but it's still a form of methionine. The Shine-Dalgarno sequence is a trigger for the ribosome to bind. It is only present in bacteria, but not eukaryotes. And in bacteria, as soon as any RNA has been transcribed, it can get translated. That is not true in the eukaryotes. I mentioned mutations in terms of the wobble in our genetic code. that we could have a mutation and still end up with the correct amino acid. So a mutation is any change in the DNA, and it may or may not lead to a change in the protein. So we are all almost certainly mutants. We have 3.5 billion nucleotides that every time there is a cell division, all 3.5 billion of them have to get copied. The chance that that has happened perfectly, the billions of times we've had to have cell division, is highly unlikely. If we're lucky, the mistakes that were made end up with wobblies and we get the right protein. The enzymes, when we do our DNA replication, also don't just make one pass like most of us do the night before an assignment. They actually go back and proofread. So if a mistake gets made, DNA polymerase can back up and reread that DNA and make sure it corrects it. But if a mutation happens, we can look at the effects of point mutations. One single base getting changed. So we might have a silent mutation. The wild type or normal DNA should give this RNA with a serine, valine, proline, and tyrosine amino acid sequence. If we change the 6th nucleotide to a T instead of an A, we get a U instead of an A, and we get serine, valine, proline, tyrosine. There is a mutation. The DNA has absolutely been changed at one position. The RNA was changed. However, the genetic code wobbled, and we got the correct amino acid. There'd be no change in the function of that cell. We could have a missense, where we change a different amino acid or a different nucleotide, and here we got the first 2 codons are correct, but because we changed a letter that was the first letter of a codon, we should have had CCC, but because of the mutation to an A, we get ACC, and now that is the amino acid threonine. We have the right number of amino acids, and everything before the mutation is okay, everything after the mutation is okay. But there is a change here, and that is called missense. It has some sense because we have the right number of amino acids, but it's a little bit off. We could have a nonsense mutation. This changing the C at the end to a G means that we end up with, instead of a UAC, which is tyrosine, an actual amino acid, we end up with UAG, which is stop. Now that makes no sense. We have cut the protein short, so that is called a nonsense mutation. And the bottom one, where my face is, there we go, move me out of the way, is what happens if we have an insertion or a deletion. Still a point mutation, one single nucleotide change. In the first one, we are adding a nucleotide. It should be AGCGTA and a CCC. Well, that's our normal one. If we remove the T and the A, that's removing 2, we could just remove 1, it messes up that codon and everything after it. Changing one nucleotide might have no effect. It might give us an amino acid, but it's not the right one. It might terminate the whole chain. If we remove a single nucleotide or we add one, it's called a frame shift because it will affect the codon where it happened and every single codon after that. It shifts the reading frame because we should be reading it 1, 2, 3, 1, 2, 3, 1, 2, 3, and instead we have 1, 2, 3, 1, 3, 3, 3. It's off. So An insertion or a deletion will absolutely always result in multiple changes in the protein, and unless it's some crazy fortuitous beneficial change, this protein, if it's functional, is not going to have the function that originally was supposed to have. Move my head again. There we go. This is a 5-year-old map of HIV prevalence, looking at people 15 to 49 years. Why all of a sudden does an HIV map show up in a chapter on DNA? HIV is a retrovirus. Its genome is actually single-stranded RNA, but it carries that enzyme reverse transcriptase. Now it might make a little more sense, the name. Transcription is reading DNA to make RNA. So reverse transcription is reading RNA to make DNA. Because of that, HIV is not that picky with exactly how its genome gets made, has a very high mutation rate. And the rate of spread can be related to the strains or the mutation form of the HIV that is present. It is also related quite highly to the availability of education in terms of prevention of HIV, the stigma of seeking help for having HIV, the availability of condoms to prevent the spread of HIV. So we can see the global prevalence is about 0.8%, but if you go to West Central Africa, East and Southern Africa, anywhere from 2.2 to 7.1%. That is an extremely high and sad rate of such a deadly disease. But that rate of spread can be related to which particular variation of the genome of the virus is present. We're looking at these different analogs of nucleotides, and we're looking at them because analogs, things that are close to the shape of a nucleotide but not quite, are one class of drugs for dealing with viruses. Antibiotics don't work. Antibiotics only work against bacteria. The name literally means against life. Viruses don't have many of those characteristics of life, so antibiotics don't work. So a normal adenine nucleoside because it doesn't have the phosphates. If the phosphates were present, it'd be a nucleotide. So there's a normal adenine. 2-aminopurine, instead of an amino group here, has a hydrogen, and instead of a hydrogen here, it has an amino. So we swap the position of those two functional groups. And that means if this analog gets incorporated into the viral genome, then it's not going to be able to hydrogen bond correctly with a thymine. And that will disrupt the replication of the viral genome. We also have cytosine in the presence of nitrous acid can be converted into a uracil, and then it will base pair with adenine. That is an incorrect base pairing, will cause high rates of mutation in the viral genome and will disrupt its replication. And there's also a guanosine analog, which instead of having the sugar be normal, the sugar gets disrupted. It has the top part looks correct, but it doesn't have the bottom part of the deoxyribose or the ribose sugar, in which case it can't continue being built into a polymer. So that means that these analogs, these structures that are similar to but not the same as a nucleotide, can be used to disrupt the viral replication. Other things that can damage DNA that may not be used therapeutically, but are chemicals we need to be aware of to be safe, can be intercalating agents. Acridine is one example. To intercalate, it sticks between the strands of the DNA and binds, and it causes disruption. It causes a wrong spacing between the base pairs. So when DNA polymerase reads this, it might make a deletion, it could have insertions. Either way, it's going to disrupt how the polymerase can read the DNA correctly. Ionizing radiation is called that because when that radiation strikes molecules, it creates ions, ionizing. When the ions are created, that is going to disrupt the bonding in the molecules. And if it disrupts the bonding in nucleic acids, it can cause breaks in that covalent bonds between the phosphates and the sugars. It might do it only in one strand. It might do it in both. Either way, broken DNA, not a good thing. And non-ionizing radiation doesn't create ions, but ultraviolet light causes thymine dimers. Let's move me out of the way again. So thymine is that nitrogenous base that's only found in DNA. A dimer means two monomers stuck together. In a normal DNA strand, the bases point inward and hydrogen bond across. They should never ever interact with each other. UV light, however, causes two thymines that are on the same strand to covalently bond with each other. And when they covalently bond, they are not hydrogen bonding with their adenines on the other strand, and this strand might get read okay by the DNA polymerase. This strand will not. DNA polymerase won't know what to do with that. It might read it as a 1 base. It might read it as, maybe that's a G, and put something across from it. And that is going to cause some manner of mutation. We do have enzymes that can break this covalent bonds that are made during thymine dimers, but if the damage of the UV light is happening at a higher rate than we can repair it, then we end up risking mutations in those cells. And that is why UV light can be cancer-causing in our skin. Our skin cells are right up there next to the light. Exposure to high levels of UV light can cause thymine dimers at a faster rate than our cells can repair it, and that can disrupt the control of the cell cycle. Move me again. There we go. So bacteria have two different ways to repair these thymine dimers. In our face-to-face lab, we definitely do this experiment. I'm hoping we will have time at the end of the semester for me to demonstrate this for you. But what we do is we slather plates with bacteria, Serratia marcescens, that turns red. And if we just incubate that plate, it should grow a nice solid mass of red bacteria. But if we put cardboard on top of that that has little holes in it, stencils, and then we stick that under UV light and bombard it, where the cardboard covered it will be protected from UV light, but where there was a hole, those bacteria get damaged by UV light. And half the plates we set on a shelf in the lab in the sunshine, so at night it's dark and during the day there's light, and half the plates we wrap up in foil, and so it will always be dark. So there are two different types. We can have nucleotide excision repair, where a enzyme will cut the strand on either side of the covalently bound thymines, cut that out, and DNA polymerase I will come in and create a new healthy strand. In photoreactivation, light, normal light, not UV light, triggers an enzyme that just cuts out the thymines, or doesn't cut them out, breaks the bond between the thymines, and so now we have a normal pair. So the nucleotide excision repair cuts out a whole chunk and replaces it. Photoreactivation just breaks the covalent bond. So in the experiment where some plates are left in the light so that they can do both photoreactivation and nucleotide excision, they recover much better than the plates that we wrap up in foil where it's dark, because they can only do nucleotide excision repair. They cannot do the photoreactivation. We don't have photoreactivation, we only have nucleotide excision repair. There are ways to identify mutants, and I'm probably going to eliminate this slide, so I will skip it. But there are ways for prokaryotes to swap genetic information. 3 different primary ways: transformation, transduction, and conjugation. Transformation means this cell is somehow able to take in DNA from its environment and incorporate it into its chromosome. This happens naturally with many species of bacteria. DNA is a huge molecule, but they have transporters in their cell walls and cell membrane to bring DNA in. Transduction means a virus, a bacteriophage, injects its DNA, and that gets incorporated into the chromosome. That would be the lysogenic cycle that we saw with the bacteriophage, where unless there is some condition that triggers it, the viral DNA stays in the bacterial chromosome and gets replicated along with bacterial chromosomal DNA. In conjugation, we have 2 bacteria. These little spikes represent the pili, which are protein projections that help the bacteria stick to surfaces, but also to each other. This bacterium has its chromosome and a plasmid, an additional circular piece of DNA. When these 2 bacteria connect via the pilus, the conjugation pilus, a copy of the plasmid is made and passed to the recipient, and when it's complete, the original cell has its chromosome and a plasmid, but the recipient cell also will have the chromosome and the plasmid. Usually it has to be the same species of bacteria for this to happen. For the conjugation, definitely. For the transduction, usually, because viruses are pretty specific as to what type of cell they infect. But for transformation, this DNA could come from anything. As long as it's a plasmid piece of DNA and gets into the cell, it could be Streptococcus taking in Streptococcus DNA, Staphylococcus taking in E. coli DNA. Remember, a free DNA molecule is chemically just DNA. So this is one way that antibiotic resistance can spread very rapidly. If the information for being resistant to the antibiotic is on DNA that's just in a cell or in a host, the bacteria can take it up, and then every time they grow and divide, they also contain that information for antibiotic resistance. There's a nice little Vibrio cholera because they produce toxins that then can be picked up by other cells. This is showing the F plasmid, which is called the fertility plasmid, getting passed via the pilus from one cell to the other. It can sometimes integrate, and then we have the F plasmid integrated, and then if it, under different conditions, can get cut back out and be separated again. It's called an HFR cell because it has a high frequency of recombination. You're not going to have to calculate how far they know the transfer happened. Transposons are jumping genes. They are a sequence of DNA that has inverted repeat sequences on either side and can cause a folding of the DNA so that instead of this DNA being before the green, it is now in the middle of the green and disrupting it. This is a naturally occurring type of jumping gene and can be a way of doing regulation. This gene is active if it's not interrupted, but if the transposon jumps in the middle of it, now it's going to be inactive. In prokaryotes, we can regulate our genes by putting related genes all together on the chromosome in a structure called an operon. So structural genes mean they encode a particular product. Gene A encodes protein A, gene B encodes protein B, etc. The promoter is that sequence to which the RNA polymerase binds to start transcription. There is also an operator. Both the promoter and the operator are sequences of DNA. The DNA sequence of the promoter tells the RNA polymerase to bind. The DNA sequence of the operator is going to allow either a activator to bind and allow things to happen, or a repressor can bind and prevent transcription from happening. We are looking at the tryptophan operon. This is a series of genes that encode the enzymes necessary to make tryptophan. So if there's no tryptophan in the environment, and tryptophan is an essential amino acid, it's necessary amino acid, then the microbe will need to make its own tryptophan. And so the RNA polymerase will bind and will read this DNA and transcribe all of the genes that are necessary to make tryptophan. But if tryptophan is present, that means the cell should not waste any energy building its own tryptophan. And the tryptophan will bind to a molecule called a repressor. That tryptophan plus repressor sticks to the operator, the switch, and even though RNA polymerase still recognizes the promoter, there's a big fat protein sitting on the operator, and we do no transcription because we don't need enzymes to make tryptophan if we can get the tryptophan for free in the media. With the lac operon, it is genes that will allow for the digestion, the breakdown, the catabolism of lactose. So in the absence of lactose, if there is no lactose to eat, we do not need the enzymes to eat lactose. And so the repressor binds to the operator, and again, even though RNA polymerase can recognize and bind the promoter, nothing happens. It gets stuck. However, if lactose is present, then the lactose pulls the repressor off of the operator, and RNA polymerase will transcribe all of the genes that the bacteria need to digest lactose. Now this has nothing to do with eating lactose or digesting lactose in humans. This is just in bacteria. We don't have operons. We don't have a collection of genes that are all under the control of one promoter. That doesn't happen. So this is just in prokaryotes. This is showing that if we have glucose and lactose present, what's going to happen with the E. coli metabolism? If both are present. In the previous one it said, well, if lactose is present, we're going to yum, 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 make enzymes to eat lactose. However, if there's glucose, E. coli is going to eat glucose first. Glucose is a single sugar. Lactose is a disaccharide. So it's going to take enzymes and energy to break the lactose apart and then the enzymes to digest those. So if glucose is present, we're going to eat the glucose first, and then there's going to be this little lag time while the cell's going, what the hell, what happened to my glucose? I'm like, oh, I've got lactose. Okay, no glucose available, but I do have lactose. This is when the cell is transcribing and translating those enzymes to digest the lactose, and then the growth can continue. Regulation can also happen based on ATP levels. So if ATP levels decrease because glucose levels have dropped, what few remaining ATP remains can be converted to cAMP, cyclic AMP. It's called that because it is adenosine monophosphate. It's cyclic because the phosphate is attached into two places. And so if there are lots of molecules of cyclic AMP, that is a cell signal that glucose levels have dropped and you need to start looking for a different food source. With regard to our lac operon, if there's no cyclic AMP around, then we don't have cap binding to the promoter. We have low level of transcription. But if we do have cyclic AMP, high cyclic AMP means low glucose. Low glucose, and we've got lactose around, we will increase the production of these enzymes. And if we have the cyclic AMP and cap getting our transcription, it'll go along great as long as lactose is present. But if the lactose is absent, it doesn't matter if we've got these two molecules bound. The repressor will block that operator. So this is looking back at our tryptophan operon. If we have lots of tryptophan, then we are not going to synthesize the trip operon. And if the trip levels are depleted, then we will synthesize the enzymes for making tryptophan. These are called ribo switches because they are RNA, which contains ribose, and it acts like a switch because folding in on the RNA is signals for molecules to bind and carry out effects on the cells. In eukaryotes, it gets really messy. We have enhancer DNA that can be way ahead of the promoter. So if this were linear, the enhancer would be all the way back here. And that enhancer, this sequence of DNA, allows for the binding of a whole bunch of proteins called transcription factors, and that's what allows the RNA polymerase to bind and continue. This is an image of translation. So you should be able to recognize that these are all codons. This is the direction of the synthesis. We've got the E site, the P site, and the A site. This is the single amino acid attached to a tRNA. This is our peptide, and be able to recognize those parts of the synthesis. I will post these slides without the video so that you can go back and look at that video for DNA replication. I have also separately provided links for DNA replication and for translation. I know that audio descriptions of still pictures are not the most useful, but combining that with those videos that have captions can really help to solidify these fairly abstract concepts.